you, Dr. Bondarenko, for this nice introduction. And I thank you and the organizing committee for inviting me to attend this interesting meeting. I'm especially honored due to the fact that I have been selected to be the first speaker on this morning. I hope I will present you something which might be interesting to you, although I think I'm a kind of exotic speaker here. I'm not in the real mainstream of metabolomics. I'm working on Institute for Chemical Ecology, and my subject will be about analytics in chemical ecology. So during my talk, I will tell you about our work, our research, and I will also present you some slides which are taken from other researchers. So I will give an overview on this field, including some work of our own group and some work coming from colleagues from other institutes working in similar field. First slide shows you the institute building. I think it's a nice environment there, and I'm happy to work in this institute. And according to the circumstance that I am a little bit exotic speaker here, I would like to introduce you a little bit more to chemical ecology and to natural products in chemical ecology. So natural products are formed by microorganisms, by plants, by animals and other beings for the benefit of the producer. So it doesn't mean that they are producing their chemicals for the benefit of mankind. And that means that they are optimized for the purpose of these organisms, and they are involved in interaction between different organisms. But it is also clear that these compounds, these natural products, have some useful properties which can be used by human beings for medical purposes, for cosmetics, nutraceuticals, and so on. And I think this is the main subject of this meeting, but I think it could be also interesting to talk a little bit about the role of these compounds for their own producers. So in chemical ecology, we are studying interactions between different organisms, and in our institute, we are focusing on the interaction between insects and plants. So the field of chemical ecology covers some research in natural history, field observations. It also covers genetic and molecular biological studies, biochemical investigations to look for enzymes and principles which are regulating enzyme mechanisms and the production of small molecules. And my area is especially the analytical chemistry of small molecules. So to identify compounds, to isolate compounds, to identify them, to do some metabolomic studies, and to localize these compounds in the tissue, and also to study the biosynthesis of natural compounds, how they are produced and how they are transferred to in the pathway. So the ecological interaction between insect and plants, or plant-plant communication, is an important field in our research. And of course, different organisms have different metabolic profile, and the profile is different, for example, in different plant parts, in the flower petals, in the green parts, or in the root systems of plants. And they are affected by environmental factors, for example, by irradiation or by chemicals or by temperature and whatever. And they are, of course, also affected by genetic modifications. And this is what we are interested in. So these compounds which are attracting or communicating or involved in communication. So this is, sorry, I will go back. This is a flower, and the yellow color is for attracting pollinators. You can see visible flowers, or you can see UV absorption, and both of them are, of course, involved in these interactions. This impressive and well-known famous picture shows the bombardier beetle attacking an enemy or responding to an attack of an enemy. And also chemicals are involved, of course. In this case, quinones are the main compounds which are toxic to or repellent to enemies of this beetle. But in many cases, the interactions are much more complex. For example, this Nicotiana attenuata, this white tobacco plant, this is producing not only nicotine, but many other compounds which are involved in direct defense, so directly repellent or toxic to other organisms, or which are involved in indirect defense. So that means attracting the enemies of the herbivore. Then we can discriminate between constitutive defense metabolites or inducible defense metabolites. And the study of the investigations on these compounds is what we are doing. As you know, there are many compounds which we now call specialized metabolites. Previously, almost denoted as secondary metabolites. Nowadays, denoted as specialized metabolites. So they are defense compounds, semiochemicals, and so on. And they are not only a small number, they are really diverse in their structure, and this is a challenge for the chemical studies in this field. So what we are doing is the complex multi-component chemical investigation of biological samples. And we are using different methods, of course, spectroscopic methods, UV spectroscopy, just looking at the electron shell, or mass spectrometry, the mass of compounds. So this slide is taken from a teaching lecture. Sorry, excuse me for that, but I think it illustrates very nicely that we are looking for different properties of the compounds using different methods. And infrared spectroscopy, also the bond vibration is important. And of course, this is my special field, NMR spectroscopy, to looking for the properties of the atomic nuclei. NMR and mass spectrometry, of course, are the most important methods in analytic methods in chemical ecology and in natural product chemistry in general. What we are doing is identification, dereplication, structural elucidation, de novo structural elucidation. We are doing some metabolomic studies, metabolic profiling, and one special field is organ, tissue, and cell-specific localization. I will be talking tomorrow about this special field. And also, we are studying biosynthetic and metabolic pathways. And we are, of course, involved in confirming the structure of synthetic compounds or of isolated natural products. And there is a trend nowadays from pure compounds, from isolated pure compounds to extracts, to fractions, mixtures. And it goes on into studies of the intact organism in organs or in tissue of plants or insects or other organisms. And in NMR, especially in NMR, we are using conventional 1D and 2D NMR methods, but we are also using coupling techniques and also the use of chemical shift imaging and imaging techniques in chemical ecology is increasing. I will start with an example of identification. So this is the flower of the Iceland poppy plant. This is interesting plant because this pigment has been investigated for many decades. So the first time it was isolated in 1939, and this example shows that in this petal there are many different compounds, and it is important to find out which compounds are really interesting. And so we investigated this plant, or the petals of this plant, and we found that there are a lot of flavonoids which are colorless, so they do not contribute to the color of the compounds. But there are other compounds which have different UV spectra. They seem to be the pigments, and according to their wavelengths or absorption wavelengths of more than 400 nanometers. And so the structural elucidation of these compounds is much more interesting. And sorry for this slide, it shows some structural elucidation, and as a chemist I cannot resist to show you also some such example. we have identified the structure of these compounds, and it was not really easy to do that because this area so you can see that there are only there's only one proton here in this area, and this makes it very difficult to identify or assign all these carbon and other atoms in this region. So we had to do some derivatization, so hydrogenation, hydrolysis, and methylation in order to introduce more protons in this area, or to make the spectrum more simple. And then we were able to identify this structure, and in cooperation with another group, also the stereochemistry of this compound, the absolute configuration in these two positions. So I will not continue to talk about structural elucidation. I think I will go to some metabolomic studies or some information about metabolics, what we are doing in metabolomics. So the question is why metabolomics or metabolic profiling is interesting for chemical ecology. What can it contribute to chemical ecology? So of course, the metabolome is part of the phenotype of an organism, but it is most important that metabolomics is a tool for understanding the metabolism and the biochemistry of an organism. And this is the reason why we are doing metabolomics in chemical ecology. So, of course, the metabolome is determined by the genome. So metabolites that are characteristic for a certain organism or for a species or cultivar. And metabolites are also changing or are different in organisms which have been modified genetically. Of course, metabolic changes are different or are coming up at different developmental stages of an organism and during circadian rhythms. And metabolites are changed in response to biotic ecological interactions, for example, as I mentioned before, interactions with other organisms and so on. So in reality, in my opinion, metabolomics in this sense doesn't exist. What we are doing in reality is metabolic profiling, because we are not yet in the situation that we are able to identify really all the metabolites in a biological system. So the more correct term would be, in my opinion, metabolic profiling. The metabolic experiment, this is well known to you, and I do not want to go into the detail here, but I would like to focus a little bit on this point, data analysis. Usually, I think you are familiar also with NMR-based metabolomics. Bucketing is used to simplify the spectrum and to analyze the data. But in recent years, there are some more advanced techniques have been introduced. For example, differential 2D NMR spectroscopy by the group of Bernd Schneider in Cornell. And they have recorded 2D spectra, cozy spectra of the wild type organism and of a genetically modified or challenged metabolism organism. And then we have subtracted those spectra, and by that way we are have been able to identify the active metabolites. Quite straightforward approach, and we are using this and establishing this in our institute now for our purposes. Some examples are in progress, but I cannot show you in the moment. But in general, metabolomics in the past means that we are determining quantitatively compounds in an organism at a given time point. So that means we are studying whole organisms. This is a kind of bulk tissue, and of course such studies result in average content of secondary metabolites or specialized metabolites in an organism. And examples are shown here. It is trivial that compounds are distributed within a plant in a different way, so that some compounds only occur in the roots and others do occur in different plant parts, and others only occur in the green part. And even cell-specific separation of compounds has been observed, for example, in these root cross-sections by the fluorescence of the compounds involved, included in the yellow cells and in the red cells. So that means a non-uniform distribution of secondary metabolites in plants is very common, and that means also that a whole organism metabolomic approach is not enough. So we need in future a tissue or cell type specific metabolomic approaches. And this is something I will talk about tomorrow, what we have done in this area. But we also need to study the metabolomics in not only in one individual of a species, but in many of them. For example, in long-living organisms like trees, such organisms, we have a certain fate. We are affected by environmental effects. They are changing the metabolic profile. It is obvious in this comparison of proton NMR spectra that there are some differences, and this is due to the changing metabolome during the lifetime of such a tree. And that means we have to study not only single individuals, but we have to compare a certain series of individuals of the same species in order to establish the metabolome. So to summarize this, we have to, in future, we have to look for organ and cell type specific metabolomics. We have to look for comparative metabolomics of individuals of a species, and of course we have to look at time-dependent metabolomics. So that means metabolic fluxes. So it's spatio-temporal metabolomic analysis or fluxomics. This is what we have to do in future. Not only in medical metabolomics, but also in chemical ecology. In the final part of the talk, I will go into detail a little bit about biosynthetic and metabolic pathways and fluxes. And I will talk about isotope labeling, and especially about the usefulness of labeling for quantification and for sensitivity enhancement. So labeling with radioactive compounds has been used in the 50s and the 60s of the last century to identify metabolic and biosynthetic pathways. And nowadays, these labeling studies are almost replaced by biochemical methods, by molecular methods. But there's a certain advance of new technology in this area, and we are using carbon-13 labeled or stable labeled isotopes, stable isotopes for labeling in order to establish pathways, fluxes, and also metabolic conversions. So already in 2004, Kikuchi from the Riken Institute in Japan proposed to use labeling of organisms, of plants, for example, with stable isotopes to enhance sensitivity, to use high field NMR, and then to improve the quality of the signal to noise of NMR spectra, for example, for HSQC spectra, to have in a shorter time a better quality of HSQC spectra, and from that to be able to quantify metabolites in the spectra in the sample. This is also a teaching slide showing that the quantification of labeling can be done by mass spectrometry, of course, but in such a case, the molecule or the fragment specific enrichment has been measured or can be measured. With NMR, the quantification of precision-specific carbon-13 enrichment is possible, and that the method development is in this area is in progress in the moment quite rapidly, and there are really interesting approaches how to quantify individual carbon-13 enrichments in complex molecules and even in complex mixtures. One approach to study biosynthetic pathways is the so-called retro-biosynthetic analysis. This has been developed and introduced mainly by Wolfgang Eisenreich in Munich University, and it uses mixtures of uniformly labeled compounds with non-labeled precursors. These mixtures are applied to plants, for example, maybe uniformly labeled glucose, fatty acids, also carbon dioxide has been used, and incubation of these compounds with the organism, with the plant, results in photosynthetic compounds in the central metabolism. And then these compounds, these central metabolites are isolated, and also the target compounds will be isolated and compared their labeling pattern, their isotopolog pattern, by carbon NMR and by mass spectrometry. And from these isotopolog patterns, it is possible to reconstruct metabolic pathways. And this is what we have done in several examples during last years, especially in an example I will show you in a minute. This shows just the pulse chase labeling experiment using carbon dioxide. So this is a non-invasive, completely non-invasive approach, because carbon dioxide is assimilated by the plant. It is metabolized. It is assimilated in the photosynthesis, and then introduced. The labels or the compounds are introduced in the central metabolism. And in the pulse phase, labeled carbon dioxide is applied for a certain time, and there's a non-random incorporation of carbon-13 into the central metabolism. And after that, after the pulse, the post-pulse or chase phase, again, non-labeled carbon dioxide is assimilated, and the pattern of metabolites can be interpreted by NMR and mass spectrometry. There are some commercially available incubation chambers available for this kind of experiments with labeled CO2. We have constructed our own one, which has a flexible bag size or container size, and which can fully control the conditions inside this chamber. So this is an example how to interpret the data in NMR spectrum, especially the carbon-carbon couplings are very important in this approach. CC couplings can be taken or can be extracted from the spectra, and then a kind of labeling pattern is established and finally integrated, and shows a quantitative pattern, isotopolog pattern of the target compound. And these compounds are then compared with primary metabolites or other metabolites in the same mixture. And now I come back to the example which I have been introducing before, the Iceland poppy flower petals, the yellow color, is nudical in the structure. The biosynthesis of this compound, of course, was not known, and what we have done here is to use this carbon dioxide labeling to compare the labeling pattern for the well-known flavonoids with the pattern for the unknown structure of, or an unknown biosynthesis of nudicalines. And from the NMR spectra we could extract the CC coupling patterns, and we could integrate the intensity of the signals and could identify these isotopologues and compare the isotopolog of the nudicalines with that of camphorol, and prove that in the nudicaline biosynthesis a plant, a pathway is involved which is identical with the flavonoid pathway for the formation of camphorol or flavonoids. And from this isotopolog analysis we can reconstruct the whole pathway, not only for the flavonoid part, but also for the indole part, which forms the other half of the molecule. So to summarize, what I have been telling you is that we are using analytical methods in chemical ecology to identify new compounds, to quantify compounds, to do analysis and measures in terms of metabolomics, metabolic profiling, and also to localize some natural products in a cell-specific manner and investigate their formation through their biosynthesis. Finally, I would like to thank my group. These are the people who have done the work. There are some people who have been cooperating, cooperation partners, mass spectrometry group in our institute, Wolfgang Eisenreich in Munich, Jonathan Gershenson in our institute, Oliver Kaiser in Dortmund University, and there are some funding organizations who gave the money. And I would like to thank again my group and all of you for attention and for being here and for listening to my talk. Thank you.